Hello, everyone, and welcome back. We've had two excellent presentations so far, and we're very excited for the next two. So I would like to welcome Sara Vazquez from the University of California at San Francisco. And for many of you, you'll recognize Sara as she attended our symposium in 2019, and we're just thrilled to learn more about her work as it relates to APS type 1. Welcome to Sara to speak about autoantibody discovery in APS type 1. Thank you.
Hi, everyone. My name is Sara Vazquez. I'm really excited to be here today to talk to you more about autoantibody discovery in APS-1. I did get to attend the 2019 symposium, which was a really wonderful experience to both meet some of you and also hear about the research. So I'm very excited for this opportunity today to present some of our recent work. I want to start by saying that most of what I'm going to talk about today is actually available in a publication that we published last year. And I've included the link here in case anyone is interested in learning more about the work that I'm going to talk about. One of the nice things about this article is it actually starts out with a few paragraphs explaining the work for non-scientists. I also want to start out by acknowledging that this work that I'm talking about today was the team effort between Mark Anderson's lab, Derisi's lab, and Michael German's lab at UCSF, and then of course the Leonakis lab, and in particular Elise was a huge help to this project. And obviously at the center of this is all of the patients and the clinicians who have contributed to this, and none of this work would have been possible without all of you. So I'm very appreciative. I want to start by giving a little bit of background on autoantibodies, why we care about finding new autoantibodies, and what autoantibodies and how we can go about finding new ones. So antibodies are large Y-shaped proteins, and they're produced and secreted into our blood by B cells, and they form a really critical part of our adaptive immune system. So one of the main purposes of antibodies is to bind and help clear pathogens like bacteria and viruses. But on occasion, our bodies accidentally can form autoantibodies. And so instead of recognizing foreign pathogens, autoantibodies recognize self, so our own cells and organs. And this can lead to damage to those cells or organs, and that can result in autoimmune disease. So autoantibodies recognize autoantigens, and that's a term that I'm going to be using frequently in this talk. An autoantigen refers to the target of the autoantibody. So for today's talk, these are all proteins, but in general, autoantigens can also be lipids or nucleic acids, and there's really a broad spectrum of things that can be targeted by autoantibodies. So why does identifying autoantigens and the specific protein targets of autoantibodies matter? So first of all, understanding exactly what is being targeted, so what cells are being targeted, what proteins are being targeted by our own immune systems can really help us better understand the mechanisms of disease. And it actually helps us to then ask downstream questions about why those antigens might become targeted. Second, autoantibodies can be stable and robust clinical markers that have potential to be used both diagnostically or predictively. And finally, if we understand better what is being targeted by our immune system, then far into the future, it's potentially possible to intervene in those very specific responses. And so better understanding exactly what's being targeted can help with that too. Why does autoantigen discovery matter specifically in APS-1? So as everyone here knows, patients with APS-1 can develop many different autoimmune manifestations over the course of their lives. And so on the left is just a drawing of some of the organ systems that can become targeted by autoimmunity in APS-1. This is actually not even all of the manifestations, it's a handful of them. And on the right, I'm showing a diagram from a paper out of the Leonakis lab a few years ago that's showing the number of total manifestations in each patient with APS-1, and you can see it can range from 2 to 14 or even more. And many of these individual autoimmune disease manifestations are still not that well understood. However, we do know that in some manifestations, specific autoantibodies correlate with diseases. So again, it helps us better understand what the immune system is targeting in those diseases and could hold future potential for diagnostic or predictive testing. And I just want to point out 2 examples of this. We know from previous publications that antibodies to SOX10 are very correlated with the disease manifestation of vitiligo, which is an autoimmune depigmentation of the skin. We also know from previous publications that autoantibodies to the cytochrome P450 family are associated with adrenal insufficiency. I also noticed that my drawing here did not include the adrenal glands, and so you'll just have to imagine them for today. However, many of the manifestations, for example, tooth enamel hypoplasia or intestinal dysfunction or nail dystrophy, are still not that well understood. And there are, for many of these, there are no known disease autoantibody associations at this point. So how do we go about finding new autoantigens? There are multiple approaches to finding new autoantigens, and I've simplified here into 2 approaches, a candidate-based approach and unbiased approaches. There are many approaches that fall somewhere in between these 2, but I wanted to briefly explain these approaches. So first of all, a candidate-based approach, also known as making an educated guess, is the way that many autoantibodies have been found. Sorry about that. And so the way that this looks is something like, we know that patient X has autoimmunity to an organ Y. We know that organ Y expresses a lot of protein Z. And so then we want to check, does patient X make autoantibodies to protein Z? So we're really testing 1-to-1. Do we see autoimmune response to this specific protein? In contrast, an unbiased approach can look something like this. We know that patient X has autoimmunity to multiple different organs. We know that all organs express a lot of different proteins. And so we want to simultaneously be able to test for antibodies to all 20,000 proteins and see what's there. And that's what I'm going to be talking about today in the form of PhIP-Seq, which I'll explain a little bit further. So PhIP-Seq stands for phage immunoprecipitation sequencing. Bacteriophage is essentially just a virus that can infect bacteria. And for today, and for this case, it's really just a great molecular biology tool because we can really easily manipulate its DNA and make a lot of them for very low cost and with very little effort. So what we're able to do is take the information for pieces of proteins and insert that into their DNA sequence so that these bacteriophage are made to express the proteins on their cell surface for display. So essentially, we're just putting all of the information for making all 20,000 proteins into these bacteriophage, and then end up with a library of bacteriophage. I want to briefly explain the experimental overview, and I'll do this once with a little bit of scientific jargon and then once simplified. But essentially, we can take this proteome-wide peptide phage display library and combine it with patient serum. Then we can immunoprecipitate the antibody phage complexes with protein AG beads and amplify those remaining phage in E. coli. We can then extract DNA, amplify and barcode, and then using next-gen sequencing and downstream analysis approaches, we can identify which phage we're interacting with an individual patient's autoantibodies. The patient serum that we use, we really require only very little for this assay, so that's a huge benefit. It only requires about 2 microliters of blood, and so that's less than a drop of blood. Simplified, we take all of these 20,000 proteins and we combine them with patient X's serum and autoantibodies, and then we're able to see which of these interact. So what proteins are these autoantibodies targeting in a given patient sample? So in summary so far, what I'm going to talk about today is combining APS-1 patient cohorts with a high throughput antibody discovery method, such that potentially in the future there could be diagnostic use as well as improved understanding of disease mechanisms. Ultimately, our goals here are to identify novel disease-associated autoantigens. For example, we want to look for autoantibodies that correlate with intestinal dysfunction, with enamel hyperplasia, with ovarian insufficiency. But before we can do that, we actually have to make sure that our method is working well for known autoantibodies in APS-1. And again, the examples I had mentioned previously here are anti-SOX10 antibodies with vitiligo, anti-cytochrome antibodies with adrenal insufficiency. So we started by looking within our data set for literature-reported APS-1 autoantigens. Now what I'm showing here is the data from that. So each of the columns that I'm showing here corresponds to a single patient sample. On the left is a cohort of 39 samples from an APS-1 cohort, and then on the right are 28 non-APS-1 control samples. And so if you look just at the first column, you can see that within a single sample, we can see multiple reactivities. So we can see signal to IL-22, to some of the interferons, to some of the cytochrome family members, IL-17, and additional antigens. And so from a single experiment, we're actually able to see many reactivities in each patient sample. This is exactly what we wanted to see, because knowing that we can detect known antigens makes us much more confident that we can find real novel antigens. And just pointing out here some of the ones that I had previously mentioned. So now that we could detect known antigens, we wanted to look for novel autoantigens in our data. And we were able to find quite a few interesting novel autoantigens. So what I'm showing here on the y-axis are the number of APS-1 samples that were positive for a given new hit. This is out of 39 samples, and so you can see that the most frequent novel antigen, RFX6, was found in 18 samples initially. Then you can see some of the known antigens, and in red are some of the novel antigens that I'm now subsequently going to discuss. Among these RFX6, ACP4, and KHDC3L. All of them were present in at least a third of the cohort, even just by PhIP-Seq. Because we are testing for all of these autoantibodies in a tube that contains 20,000 proteins, we don't always know exactly what's going on in that tube. And so we always want to do a secondary assay to make sure that what we're seeing in the initial screen actually validates at the single protein level. And so that's what I'm showing here in the lower panel. We took each of these really interesting hits. We made the protein for each one of them, and we incubated it with patient antibodies and made sure that we still saw signal. In all of the cases we did, and we were actually able to pick up even more positive samples in each of the cases. So in the end, some of them were extremely common in the cohort. We actually ended up finding anti-RFX6 antibodies in over 65% of the cohort, ACP4 antibodies in over 50% of the cohort, and then KHDC3L antibodies in over 25% of the cohort. So really the next important question that we want to ask here are any of these novel autoantigens actually associated with a specific disease or phenotype? And to do this, we aggregated the data from all 39 APS-1 samples that we looked at. We looked at all of the hits that were common to multiple patients with APS-1 versus all of the disease manifestations that we had data on. And this includes, there's many abbreviations here, but just to give a sampling, this includes nail dystrophy, alopecia, hypoparathyroidism, keratoconjunctivitis, intestinal dysfunction, and many others. And what we looked for in this data is whether any of the hits correlated with any of the specific disease manifestations. Everywhere that you can see a darker colored square indicates that there is a significant association between that hit and the phenotype of interest. And so very reassuringly, right at the top, you can see that in our data, cytochrome 11A1 does have a strong clinical association with adrenal insufficiency, as we would expect from previous work. Excitingly, there are also many novel associations, and we decided to further investigate some of these. And the ones that I want to point out here specifically are KHDC3L with primary ovarian insufficiency and RFX6 with the diarrheal type of intestinal dysfunction. So in summary, for this first part of the talk, I just want to highlight that we're able to detect known APS-1 antigens with PhIP-Seq. We can detect some novel APS-1 antigens with PhIP-Seq. We find that the novel antigens are actually reproducible in additional experiments and assays at the whole protein level. And so we feel confident in those reactivities. And some of these novel autoantigens, including RFX6 and KHDC3L, have disease associations in our data. And so with that, I want to go into the second part where I talk a little bit more about some of the disease associations that we saw and a little bit of background on these protein autoantigen targets. So I'd like to start with intestinal dysfunction, which is a very common but still somewhat poorly understood APS-1 manifestation. It's present in over 80% of patients with APS-1. Again, taken here from the really wonderful paper out of the Leonakis lab in 2016. And there have been multiple proposed etiologies of intestinal dysfunction, and probably intestinal dysfunction contains many different etiologies. But one of the proposed mechanisms is a loss of enteroendocrine cells in the intestine. So in 2012, there is this very interesting paper that looked at intestinal biopsies from 4 patients with APS-1. And what I'm showing in the top panel here is that you can see in red enteroendocrine cells staining. Okay, so all of those red cells are specifically enteroendocrine cells in a non-APS-1 intestine. But in this paper, they actually showed that some of the APS-1 patients did not have any of these red cells detectable. So there was a loss of enteroendocrine cells in some patients. So what are intestinal enteroendocrine cells? On the left, I'm showing a schematic of all of the different cell types that you can see in the intestine. They all have their own functions and are really important for intestinal homeostasis. But the enteroendocrine cell is a very rare specialized cell, and it's essentially the endocrine cell of the intestine. It secretes peptides and hormones, and it's involved in a lot of different functions like gut motility, digestion, appetite, and metabolism, among other things. And there's a lot of literature showing that defects in these cells can actually cause various types of intestinal dysfunction. So we decided to investigate the relationship of RFX6 with intestinal enteroendocrine cells. And as it turns out, there's a huge body of literature showing that RFX6 is expressed pretty specifically in enteroendocrine cells in the intestine. On the left, what I'm showing is single cell data from mice. So each of those gray dots on the left is a single cell. And then we can look at the expression of all genes within each of those cells. So I'm showing an excerpt here that we really only see RFX6 expression in the intestine in a very small set of cells, and that co-localizes with chromogranin A, which is an enteroendocrine cell marker. Really interestingly, we found in the literature that mice and humans who are born with mutations in RFX6 have enteroendocrine cell deficiency, and they have intestinal malabsorption. And interestingly, mice and humans that actually develop acquired enteroendocrine deficiency sometime after birth tend to have this chronic malabsorptive diarrhea phenotype. And so all of this information together really suggests that the autoantibodies targeting RFX6 could be related to this intestinal dysfunction phenotype. So this is our data showing anti-RFX6 positive samples within the cohort. And what you can see is that the majority of patients who have anti-RFX6 antibodies also have the diarrheal type of intestinal dysfunction relative to no intestinal dysfunction or the constipation type. So this is a really specific association. There have been reports of other intestinal antigens in APS-1, but when we looked at some of these, like TPH1, it's much less specific to the diarrheal subtype and more broadly distributed across general intestinal dysfunction. And then in this final experiment, which I think is really interesting, we looked at the staining of APS-1 serum on intestinal biopsies. And so these are slices of human intestine, and all of them were stained with antibodies to chromogranin A to mark the enteroendocrine cells, so we know what we're looking at. In the first panel, we used a non-APS-1 control serum to stain that same tissue, and we didn't see any staining. So there's no antibodies in that sample that are reacting to enteroendocrine cells or any part of the gut. In the second panel is an APS-1 sample that does not have anti-RFX6 antibodies, and again, we don't really see any staining of those red enteroendocrine cells. But in the last panel, we looked at a sample, an APS-1 patient sample that had anti-RFX6 antibodies. And we see this perfect co-staining of anti-chromogranin positive enteroendocrine cells with potential autoantibodies within that sample. So this was really strong evidence that there's this specific autoimmunity in some cases targeting the enteroendocrine cells of the gut. I want to talk about primary ovarian insufficiency as well as one of the other interesting targets that we came across in our screen. So primary ovarian insufficiency is also a very common phenotype. Up to 60% of females with APS-1 can progress to this early menopause-like state, which is defined as onset before the age of 40. Very interestingly, this hit that we got from the PhIP-Seq screen, KHDC3L, happens to be very highly expressed in the female ovary and specifically within the germ cells, the oocytes of the ovary. Interestingly, KHDC3L is known to interact molecularly with NLRP5, and NLRP5 is a known autoantigen in parathyroid autoimmunity and APS-1. So when you look at the expression of these two proteins, you can see that NLRP5 is specifically expressed in two organs, the ovary and the parathyroid, and it's associated primarily with parathyroid autoimmunity. In contrast, KHDC3L is really only expressed within the ovary and the oocytes specifically. And so our hope was here that potentially we've identified an autoantibody that is more specific for ovarian insufficiency than NLRP5. What I'm showing here is, again, single cell data so that we can understand a little bit better where exactly this protein is expressed in the ovary. And so the blue grouping of cells are supporting cells of the ovary. They're basically everything surrounding the growing germ cells, oocytes, whereas the cells in the red grouping are the oocytes themselves. And what we see is that both KHDC3L and NLRP5 are really specifically expressed in the oocytes of the ovary and not within these supporting cells. So it's a very specific cell type that is being targeted here. Not surprisingly, we found that antibodies to KHDC3L and antibodies to NLRP5 tend to correlate. So patients that tend to have one of these antibodies tend to also have the other. This is not 100% true, but it's a pretty good correlation. And we also found that both of these autoantibodies are much more prevalent in females in our cohort relative to males. Finally, I think this is one of the most interesting slides for anti-KHDC3L antibodies. We looked at, for both KHDC3L and NLRP5, where different patient samples fell in terms of positive or negative. And so anything to the right of the vertical dotted line is positive for anti-KHDC3L antibodies. Anything that's above the dotted line is also positive for NLRP5. And so you can see there's many samples that are positive for both NLRP5 and KHDC3L antibodies. But pretty strikingly, every single sample within this cohort that has a diagnosis of primary ovarian insufficiency was also positive for KHDC3L antibodies. So KHDC3L antibodies were 100% sensitive in our study for primary ovarian insufficiency. One thing that you'll notice right away here is that there are additional female patients who don't have diagnosed ovarian insufficiency who have these autoantibodies. And this is something that we definitely want to study more and follow over time. We don't know potentially if this is just non-specific or if potentially it might even have some predictive effect for future disease. Either way, it will require more study, but I think it's a really interesting observation to see that there are some young female patients who do have these autoantibodies as well. I want to end very briefly with just one slide about a third phenotype that was very interesting. That is dental enamel hypoplasia. Again, very common manifestation in APS-1. I think 85% or more of patients have some form of this. And in our data, we saw a trend towards those patients with positive anti-ACP4 antibodies also having enamel hypoplasia. And so these data were not quite as conclusive as the other two, but certainly there's a strong enrichment for anti-ACP4 antibodies in patients with dental enamel hypoplasia. And one of the main reasons that we thought this was a really interesting observation is that, again, genetic mutations in this same gene cause a form of enamel hypoplasia. And so there's pretty good reason to believe that this phenotype might be related to this autoantibody in some way. So in summary, for the second part, I've discussed that anti-RFX6 antibodies are associated specifically with the diarrheal subtype of intestinal dysfunction. Something I didn't have time to talk about today is that other novel antigens that we identified, including NKX6-3 and GIP, are also expressed in the intestine, and they could add to this picture of intestinal autoimmunity in the future. Anti-KHDC3L antibodies are associated in our data set with primary ovarian insufficiency. And as I mentioned, more clinical work will definitely be needed to determine whether these could have predictive or diagnostic value in the future. They do so far seem to have greater sensitivity for ovarian insufficiency than the known NLRP5 antibodies. And finally, anti-ACP4 antibodies might be associated with dental enamel hypoplasia. And we're very interested to look at this in larger cohorts of the disease as well. I want to end with just 2 or 3 slides on what we're working on now. One of the limitations of our assay is that we were limited in number of samples we could run at once. And so recently we've begun scaling this and coming up with a protocol that allows us to run many, many samples at the same time. We've actually been able to apply this to larger cohorts of APS-1 at this point, and this is ongoing work, so I'm not going to show a lot of data here. But we looked in 3 separate cohorts of APS-1, and we're actually able to find all of the antigens that I just described across all 3 cohorts of patients. And so that makes us more confident that these are broadly specific for APS-1. And finally, there are other multi-organ autoimmune syndromes that would be very interesting to look at by PhIP-Seq, and we actually began by looking at a somewhat related disease, IPEX, where you have impaired peripheral tolerance in T regulatory cells. And in this disease too, we've started to see some potential autoantigens that are expressed in the intestine. And so it'll be very interesting to see whether these intestinal dysfunction phenotypes have any relationship to the one seen in APS-1. And so comparing these will be really informative going forward. So I'd like to thank a lot of people. Many people, more than are even on this slide, have been involved with this work, but in particular, Joe Derisi and Mark Anderson have been amazing mentors and supported this work. And then Mihalis Leonakis and Elise here have also been amazing collaborators. There are many more people, and of course, the APS-1 Foundation and all of the patients and families who contributed to this research, because none of it would have been possible without them. Thank you.
Thank you. Thank you so much, Sara. That was a really, really interesting presentation. And I have to say that I love the diagrams on your slide. I think they are so effective in communicating your work. Thank you for including them. You know, this work is so important, and I'll let people start to put questions in the chat. But just first of all, you've talked about some of the mysteries of certain manifestations of APS type 1 and how important your work could be to unlocking those connections, those mysteries as to why people experience those manifestations. Are you thinking about other manifestations also? What might the next steps be?
Yeah, that's a great question. So one of the reasons that we really wanted to scale our experiments and look in larger cohorts of patients is because some manifestations are very rare. And so we've been underpowered so far to really look for clinical associations between the hits in our screen and the clinical manifestations. So I don't have an answer yet specifically, but hopefully we'll be able to look at more of the disease manifestations that are less common than intestinal dysfunction or enamel hypoplasia in these new data sets.
Yeah, and I have to say you picked good ones to begin with, as we know from our patients and family members. Intestinal issues are overwhelming, as well as the enamel dysplasia and dental issues for our patients. So thank you for that. In terms of what the next steps may be in developing clinical treatments for these manifestations, what do you anticipate the next steps might be?
That's a great question. I do think making sure that we see these associations in additional cohorts is sort of a first step. But as I mentioned, there are potentially a lot of different etiologies of intestinal dysfunction, for example. And so potentially being able to use these autoantibodies to determine whether you might be dealing with a more autoimmune phenotype or something that's potentially more infectious disease related could be, I think, of clinical use and potentially help guide treatment approaches as well.
Thank you. Todd Tallarico has added a similar point to that question. What do you see are opportunities for our community short-term versus long-term? And what road may be the shortest? How do we get to clinical treatments more quickly?
That's a great question. I am not entirely sure at the moment. I think that before these could become diagnostic tests, they definitely need to be validated across additional cohorts. And so that's kind of what we're working on now. I think long-term, it really helps to also look for these autoantigens in additional sporadic forms of disease. Unfortunately, I think as everyone knows here, this is a very rare disease. And so I think examples from type 1 diabetes where we see crossover of autoantibodies between sporadic type 1 diabetes and diabetes in APS-1 has made those clinical tests much more available relative to the ones that we see only in APS-1. So one of the things we've been very curious to look at is whether we can see anti-KHDC3L antibodies in sporadic ovarian insufficiency or anti-RFX6 antibodies in intestinal dysfunction. And I think that by looking in other diseases, that could actually bring more attention to something that might be helpful in APS-1.
Thank you so much. I mean, we always appreciate those links with other diseases and with other research. Dr. Deal has a question and a compliment. This is beautiful talk. Have you had a chance to query the reactions of parents when their child's sample is positive for an autoantibody that is associated with a condition that they don't have yet? What have you learned about counseling families when giving results?
Thank you so much. We don't have a lot of experience with this yet in these autoantibodies that I've discussed. And part of that is because we don't really know the significance of having those autoantibodies. So even if you look in common diseases like type 1 diabetes, many individuals who do not have type 1 diabetes still have autoantibodies. And so we know that they don't correlate one-to-one with disease. And so we can't really draw conclusions if you just have an autoantibody. It might still be that you never go on to get that associated disease. It's just potentially a higher risk of eventually developing it. And so because of that, it's still completely in the research stages, not in the clinical stages, given that we don't know if any of the patients who are positive might go on to get that phenotype. So I think that's also a matter of time. We hope to study this over time to see if it could be predictive.
Thank you. And I think it's a really good point. We will have to learn how to educate clinicians and families on the meaning of a positive result.
Yeah.
We have a very important question, which is how can the patient community help your work?
I think you are already doing an amazing job of helping this work. Again, we're working really closely with multiple cohorts, but especially with the Leonakis lab. And so again, having this detailed clinical phenotyping along with patient samples is a very, very valuable resource for this research. And so in this case, I would say continue with that, because the more patients that we have there, the more correlations we can look for. And I know you mentioned initially that the registry is growing, and I think that will feed directly into this.
Thank you so much. And we have put the link for the registry in the chat for anyone who is interested on the patient or on the research side. And I can speak for the foundation, I think, and our families, that if you ever need any support, we are here to support your work, as well as the other researchers who are participating today.
So much.
I have another question, which is, do you think this is affecting the pancreas directly or indirectly when GAD65 autoantibodies have been identified?
That's a great question. And I didn't really touch on any of this today, but autoantibodies are difficult because in many cases, we don't really know if they have a functional effect or if they're just a bystander effect. And in diabetes, we have a pretty good indication that a lot of times the autoantibodies themselves are not causing that much direct harm, and they might be just a bystander effect of what's going on with the T cells. And so that's why I've kind of referred to these autoantibodies as potential markers, not as something causing the disease. Having said that, Paul and Dr. Leonakis gave really amazing talks talking about anti-interferon antibodies this morning, and those do have direct functional effects of binding interferon. So it's possible that some of these new targets, especially if they're extracellular or really available for antibodies to bind to, might have some sort of functional effect. We don't have a clear answer for that yet, but it's something that we're thinking about going forward.
Thank you so much. I think those are all of the questions we have today. Sara Vazquez, we're so grateful for your time, for your work, and that you've shared it so clearly with us today. We wish you all the best in your future work, and we are here to support it going forward.
Thank you so much.